Hey guys, so we're gonna quickly talk about transcription today. So, what is transcription? How does it work? Well, we have a template strand that we then use to make mRNA. So remember when we're doing this, we have something called a template strand and a coding strand, and your template strand makes the coding strand, aka your template strand codes for the coding strand. Okay. So that's used to make your mRNA. Then we have RNA polymerase that binds to the Tata box in the promoter region. Just remember, if you're reading a string of codons, right? If you see TATA, you should think, okay, that's the start site of an RNA. We synthesize 5 to 3 prime. There is no primer needed. So we have some very important post-transcriptional modifications. There we go, Joel. We have the 5 prime cap, which is just a modified guanine that is at the 5 prime end of your DNA, and it's there for stability plus ribosomal binding. So next we have poly A tail, which prevents degradation of your DNA. Then we have splicing. Splicing removes introns which are not active. Okay, these genes are not active. We have exons, which exons are transcriptionally active. Okay, we use something called a spliceosome, which cuts out the introns, and we only keep the exons. Okay, we can do something called alternative splicing, which is a pretty important term for your test. So, meaning that multiple proteins can form one gene. Meaning that if we had something like this, let's say we had something like this, right? And then I'm just gonna say I E1, I E2, I E3. Okay, so we're first gonna, right, we get rid of the introns. And then you could have E1, E2, E3, or you could have multiple proteins from one gene, meaning that maybe we have E1 separated, right? So maybe we have E1 connected to E2, and then we could, sorry, let's add. This is such a long analogy. But then pretty much E3 and E4 can be on their own. Okay, so that's the idea, is that you can make multiple proteins from one gene. Okay, rough example, I know. I hope that you get the bigger picture though. Okay, like, just know, okay, we can create pairs of different introns, and pairs of different introns you can have multiple different proteins, and you don't need more genes. Okay, if that didn't make sense, just comment below. Thank you. Okay, so next we're going to go over translation. Okay, I'll see you guys there.